as non-self. Essentially, your immune system is attacking your own tissue. And so this is an excessive response. Along with these theories, there's 4 of them. You also are going to have factors that are contributing to autoimmune responses. And so they're kind of separate. I mean, a big one here is mast cells. When we typically think of an excessive immune response, we're also going to have inflammation. And inflammation is strongly driven by those basophils releasing histamine, all right? And then we're going to further explore this excessive response in these things that are called hypersensitivities. And there's 4 of those as well. We'll see if we can get to the deficiencies, because I'm not going to talk that much about the deficiencies. So these are, we're really looking at an excessive response. Now, in regards to hypersensitivities, that isn't always going to mean autoimmunity, actually. Can you think of a hypersensitivity that's not an autoimmune response?
Allergies.
Allergies, exactly. That is just your body thinking pollen is a non-self antigen and having this excessive response. And in that, these hypersensitivities start destroying tissue when they really shouldn't be. They're having this abnormal excessive response when they shouldn't have to do that. They're thinking that something that is benign, in a sense, is a threat to the body. Now, the 4 theories of why autoimmunity occurs, and so these are just theories. And so they're trying to understand why our body would attack itself. And one of those is this thing called, so these are the 4 theories. The first is this thing called antigenic mimicry. Are you guys familiar with mimicry, that term? Mimicry means that something is, sort of has a mask or a disguise. So, for example, butterflies on their wings will mimicry the eyes of a predator so that a predator will not try to eat it because it'll see the butterfly itself as a predator. That's mimicry. So it's, in a sense, pretending to be something else. All right? So that's what is going on here. So the thing that is going to pretend to be something else or look like something else is going to be a non-self.
It's like a foreign epitope?
Yes. A non-self antigen will look similar to a self antigen. And our immune system, the adaptive immune response, will recognize that, but since it has a similar structure as self molecules, our body will start attacking self cells and self molecules. So that's one theory. Another one is release of sequestered antigens. And when I'm talking about this, I'm talking about self antigens, okay? And when I mean sequestered, I mean these things are isolated. And they're isolated during what process, do you think? does a B cell and a T cell, what do they have to go through to be able to distinguish between something that is non-self and self?
Over T cells?
MHC2.
Let's think at the very, very beginning, okay? They're going to be held up in either the bone marrow, if we're talking about a B cell, or they're going to be held up in the thymus, if we're talking about a T cell. What are they doing there?
Differentiate. They're gaining either an antigen or they're going to be a naive cell.
Right, so Moises is saying differentiating. And I think that's a good term here because what they're really doing is taking a test. They're becoming competent in what is self, is really what they're doing here in those areas. And so you're going to actually have some antigens that those B cells and T cells are never going to know are self because they've been isolated during this process. Does that make sense to everybody? So release of sequestered antigens during immunocompetence. Now, one area, like a lot of these things, think of where you have a blood-brain barrier. And in particular, you have in men, in the testes, they actually have a blood-brain barrier with those testes and the developing sperm in them. If they ended up having an injury there that compromised that blood testy, sorry, not brain. Where's your brain today? Down here? No. Blood testy barrier. Sorry about that. And if that barrier is compromised and those isolated cells go into circulation, your immune system is going to recognize it as non-self and have an autoimmune response. So that's another theory here. Okay? Everybody good with that one?
So basically, they haven't had the training yet to recognize what is self before
Yeah.
it gets out into the system and starts attacking everything.
Yes.
Okay.
Exactly. And I mean, these are theories, mind you. And in regards to the autoimmune response, it probably depends upon the actual mechanism that's involved in that autoimmune response.
So it's based off of context.
Right. And potentially it could vary from one person to another in regards to what it is really happening to cause that autoimmune response in that particular person. The third one is in regards to the T cells themselves. And this one is referring to when achieving immunocompetence, the thymus itself might be dysfunctional. All right, and the other thing with this theory is twofold. Or maybe not twofold, it could be one or the other. So you can have a thymus that's dysfunctional and is really not making it to where these T cells are able to properly recognize self.
So this is in like a younger person, right, who still has
Yes.
the thymus? Okay.
Yes, because this immunocompetence occurs at a young age.
Okay.
By the time we become adults,
It's gone pretty much.
yeah, our T cells and B cells have the ability to know what is self, okay? The other thing is these T cells might actually not achieve immunocompetence in the thymus, so they're doing it outside of the thymus. And if this happens, then they're not getting that guidance from cells of the thymus to fully be able to pass this test and be functional in their ability to recognize self. Okay? And then the fourth one is dealing with B cells. And in this case, the theory is that B cells aren't properly suppressed. is that in any function that our body has, there's a checks and balances. And you do want these things to stop at some point. Just like whenever you are starting to form a fiber mesh, you want that thing to stop, or else you'll just be making more and more fiber mesh and you'll block off the pathway of blood in that blood vessel. Same thing with here. At some point, you want these B cells to stop having this response. But if they aren't properly suppressed, then they're in an active mode, and it's an excessive thing that could lead to B cells starting to cause damage to tissues, or the products of B cells.
So would you say B cells are doing their job too well?
Right. In this situation, they're doing their job too well because another thing in our body didn't tell them to stop doing their job. Okay?
So is the B cell or the plasma cell that's actually causing the problem?
It's the plasma cell.
And just
Exactly.
overproduction of
Because of
antibodies attaching to something that shouldn't be?
Or they're just continuing to make more and more antibodies. I mean, honestly, once a plasma cell is made antibodies, those antibodies will be circulating within your bloodstream. At an early age, for example, you're going to be feeding off of your mom's breast milk. And just normal foods that we eat, or even the blood that goes from mom to baby, the molecules in foods that we eat actually mimics B antigens and A antigens. And babies, by the time they're born or shortly thereafter, already have antibodies for the particular blood type that they don't have. Okay? So those things can stick around. All right? Other questions? All right. These are the theories. There are things that are going to factor in autoimmunity, but they're not these ideas of why. We just know that they play a role in autoimmunity. And one of these is genetics, and in particular, people who have mutations of genes that code for MHC molecules. They've seen that that can actually play a role in a person having an autoimmune disorder. The other thing that's going to factor into any autoimmunity response, a lot of times, are these mast cells. These are basocells that release these vasoactive chemicals that are going to lead to inflammation. And we already know that inflammation itself can do damage to tissue. And then the other one is the environment. And in particular, if the environment causes you to have stress in your life. And really what's going on there is that when you're in a more stressed state, your body is making more cytokines. And if your body's making more cytokines, then that's going to lead to activation of these immune cells. Okay? The other thing is viruses. So viruses in your body or viruses outside of your body can come in and insert their genome into our cells and potentially cause somewhat of an autoimmune response. So those are the factors. These are the theories. Now what I'm going to, no questions there? So now what I'm going to do is I'm going to do an outline of the 4 different hypersensitivities over there so we can kind of compare all of them. And then I'm going to go through each one, the mechanism, because there's a lot of parts to it, all right? And I kind of think that if we have something to bounce off of, right away, it could be a nice steady thing to always reference here. So there's 4 of these hypersensitivities. And they're called hypersensitivity 1, hypersensitivity 2, hypersensitivity 3, and hypersensitivity 4. So these are hypersensitivities I have here. Now this first one is with allergies. This one here is tissue specific. This one, these two here are very interesting that they aren't always, there's some variation. And so when we look at what's going on in each of these hypersensitivities, we're really looking at the mechanism involved, all right? We can put these generalizations onto them, but there will be exceptions, especially when we're going to this area. But usually for 3, we're talking about a persistent infection. And this one's sort of an allergy too, but we like to predominantly look at it whenever we have some type of dermatitis, if you will, like contact dermatitis is a good example of this one. And so I'm gonna put contact dermatitis here because that's typically the one that we refer to when we're talking about hypersensitivity 4.
So like poison ivy?
I have to admit, I'm not entirely sure if poison ivy is it, but perhaps, but a lot of us have that. So we're thinking of a hypersensitive thing. Nickel, like being allergic to nickel, for example, and people who have a reaction with those earrings is a contact dermatitis. Poison ivy might be correct too because plant oils are able to have this, and poison ivy, I'm sure that the mechanism involved, I would guess, is what we're going to see here. All right. Now, these two are pretty quick. They're going to happen, this response is going to happen within 15 to 30 minutes. 15 to 30 minutes, you get manifestations. Persistent infection is, you gotta realize this is, you have an infection. But what's going on with this one is you're not able to get rid of it. So you're gonna start to have this hypersensitivity response as, it's gonna take longer, obviously. It's once you can't get rid of that infection, you're gonna start to see the body is starting to attack its own tissues because of this persistent infection. So it has a long onset because of that. And then the contact dermatitis, this one doesn't happen right away. But usually you'll start to see the manifestations after 24 hours of exposure. I don't know if I used to like have to have like silver earrings. I couldn't have the ones that they pierced your ears with. And the first time I got them pierced, I didn't know that until like a day or two later, and I had this red swollen area where I had my ear pierced because this was happening. So this, 24 hours or more from exposure, you start to see manifestations. Now, when we're talking about each one of these, they like to talk about mediators and effectors. And I don't want you to start to get too bogged down as you're reading, because I found the book to be a little bit confusing with this. But remember what mediators do. They're going to be some kind of molecule or chemical that's going to be an intermediate that leads to a process. Okay, so think of that as a mediator. Now, effector, you already are familiar with that term in regards to the homeostatic mechanism, right? So the effector, think of an effector for the most part as the cell that is going to make it to where you have this response, okay? So in general, think of effectors as cells, think of mediators as the chemicals that are a part of the process, all right? Now, the mediator here is going to be this thing called immunoglobulin E, and that is essentially an antibody, all right? So immunoglobulin E is an antibody. And this particular, it's an antibody that plasma cells make, okay? You have several different types of immunoglobulins. The mediator here and the mediator here are complement proteins. Okay? And we already recognize complement proteins as mediators. Now this one's the strange one. Okay, the mediator here is not an actual molecule, it's a cell. So this is the funky one. The mediator here is going to be a T cell. But a T cell is very important to having this process occur, this response, all right? And I like to think of this T cell as potentially also being an effector, to tell you the truth. Once we go through that whole mechanism, we'll look at it. But I don't want to have any kind of ambiguity with these answers, because I think these are really difficult mechanisms to get down, all right? Now the last thing are these effector cells. When we're talking about allergies, histamine is a big chemical involved in this mechanism. So what do you think the effector cell is here? A basophil. And they call it a mast cell, and that's just a basophil that's in the tissue, okay? So basophil, or mast cell, is the effector cell. The effector here is a lot of things, but I'm going to just focus on some stuff. And macrophages are a big effector in this. You're going to see neutrophils are an effector here as well. You could also, it's those cells, like NK cells could be effectors here, but I'm going to focus on macrophages, okay? But you have a lot of effectors with this type 2 tissue specific. And then the effector here is going to be a neutrophil. That's the big effector. Mast cells are going to play a role here as well. But the big effector here is going to be this neutrophil. And macrophages are the effector here. Along with mast cells, depending upon specifically what we're talking about. All right? So mast cells play a huge role. And that's because they play a big role in inflammation and that excessive response to something. guys good with that? Okay. So I'm going to first talk about hypersensitivity 1 and what happens there. Alright, so I'm going to do a little diagram and talk about all the steps that are occurring. And so what we're going to see first off is a grain of pollen, right? Because this is an allergy one, so this is what I'm using as my example, a grain of pollen. And in this situation, this grain of pollen, we're going to have a B cell that is going to recognize that grain of pollen as non-self, okay? And when that B cell recognizes that grain of pollen as non-self, it's going to eventually, through all those processes, get to a point where it's going to divide and make plasma cells and a memory cell. And the plasma cells are going to make what?
Antibodies.
Antibodies that are specific to this grain of pollen. All right? And you'll see that the next step is you're going to have these antibodies in the plasma. Okay? And what is going to happen is you're going to have a mast cell. So remember, these guys here, what is this? These antibodies, are they the mediator? Are they the effector? They're the mediator. If we look at our chart there, mediator is this immunoglobulin, this antibody. So this here is going to be that immunoglobulin E, okay, that particular form of an antibody. It's a necessary thing that has to occur to eventually lead to this hypersensitivity response. Then you're going to have this mast cell here that is going to have receptors on it. So it's going to have all these little receptors all along it. And this is in the plasma, and it's going to bind to that receptor, okay? So you're going to see that immunoglobulin E binds to a receptor on a mast cell, okay? Everybody still good with that? Now, this is a mast cell. This isn't a B cell. This is different. This is a basocell, okay? Mast cells are granulocytes, so they are going to have all these granules in their cell. And these granules are going to have vasoactive chemicals, all right? And when this immunoglobulin that's bound to this receptor of the mast cell then binds to this pollen, you now have an antibody-antigen complex, right? We're already familiar in part to some of this, okay? And this binding of the pollen to the antibody that's bound to the receptor is going to cause these vesicles that are in this mast cell to release vasoactive chemicals. And a big one, of course, is histamine. And then that's going to cause what to happen?
Inflammation?
Inflammation. And when we think of allergies, we think of inflammation, typically. So this is going to lead to inflammation. And this happens quickly. This can, from you having contact with that pollen or that allergen, this can happen in 15 to 30 minutes of that. Now, some people have mild allergies, they can have intermediate allergies, they can have severe allergies. And you're going to have different manifestations occurring here. So you can have a mild response, and in that case, the mild response, hives are actually a mild response here. You could get eczema, you could have mild bronchoconstriction. You can also have what's called rhinitis. Anything with itis, what does that mean?
Inflammation?
Inflammation, and rhinitis is meaning inflammation of your mucosa of your nasal cavity.
So this would be an example of like an extrinsic asthma attack?
A what?
Extrinsic, like allergic?
Yes, yes, yes. Yeah, because the thing that's triggering it is
The pollen.
the pollen outside the body.
Okay.
Okay? And then you can have an intermediate response. An intermediate response is going to be more severe than that, like tightening of the throat. Wheezing. You could increase your heart rate in an intermediate response. And so these things, yes, while they're mildly irritating, right? This here, you're actually going to be having difficulty getting air to that place where you want gas exchange to occur.
Because the airway's being
inflammation
closed?
is more severe.
It's because the airway's being closed that's causing the wheezing?
And also since, I mean, I would say the tachycardia, you're increasing your heart rate.
To get
so you
more
can-
air?
To get
Some more
more
blood going?
blood to the tissues.
Okay.
Yeah. And then the severe one is anaphylaxis. And what this is like, okay, here, and it's irritating. Here, I'm having a little bit of problems getting oxygen and getting stuff to my tissues. Here, you have a swollen tongue, a swollen throat. You're not getting any oxygen. Yeah, you need an EpiPen, because what an EpiPen will do is cause vasodilation, or, I'm sorry, bronchodilation. It'll open up those airways. Very good with this hypersensitivity one. All right. Now, the next one is tissue specific. And I'm going to give you some examples of that in regards to the mechanism that's going on. Yes, so this one here was hypersensitivity 1. Let me put that down. Did you take a picture? Let me put one and you can take
I got it, thank
You're good? Okay, so this is hypersensitivity 1. Let me write that down if anybody wants to take a picture. This is the mechanism of that, all right? Now I'm going to show you a couple of mechanisms with tissue specific. Can you give me examples of something that might be a hypersensitivity that is tissue specific?
RH negative mother, RH positive baby.
Right, and that's specific to that tissue. So we're not having some kind of generalized effect here, it's blood that we're talking about. So would blood transfusions, wrong blood transfusions fit into hypersensitivity too?
Yes.
Yeah, because the tissue specific here is the blood. What about a
Transplants?
disease that attacks the thyroid? Is that tissue specific?
Yes.
Yes, because it's specifically attacking just the thyroid. So when I say tissue specific, it means it's specific to a tissue type. All right? And I'm going to talk about one that you might be familiar with first, and then I'll give you a second example. The first one is, so this is 2 that I'm talking about, is wrong blood transfusion. And I'm talking about ABO grouping. So let's say you have a person that has type A blood. Okay, so this is what their red blood cells look like. And they have an A antigen on the surface of their red blood cells. This particular individual, what kind of antibodies do they have in their blood?
B.
Anti-B antibodies. That means it's against B antigens. So this is a type A individual. We're going to say this is the recipient. And they are going to have in the plasma of their blood anti-B antibodies. All right? Now, type A recipient is given accidentally, of course. This is really rare. This doesn't really happen anymore. They spend a lot of time to prevent this. But it is given accidentally type B. Now, when you're doing blood transfusions, for most cases, it's the red blood cells that you're getting as a recipient. The time that's different is stuff like open heart surgery. They'll give you whole blood in the transfusion. So you give this person a red blood cell that looks like this. All right? This B antigen here, this type A individual doesn't have the genes that code for that particular molecule. So they're going to view this B antigen as non-self. And they already have in their plasma, because of what I had said earlier, the things that we eat are going to look very similar to these molecules. And so very early on in our life, we already have in our blood these antibodies, given whatever blood type we are. Now this is going to attach this antibody, and it's going to form an antibody-antigen complex again. It's all about the antibody-antigen complex with a lot of these. And now, we're going to see when that happens, forming this antibody-antigen complex. What happens? What do we call this? What's going on with
Agglutination?
these red blood cells?
Agglutination.
Agglutination. That's clumping. That's part of that humoral response. All right? This is a hypersensitivity because you're trying to give that blood to that person so that they can function better. But it's going to have this hypersensitivity response that actually makes the situation worse for that individual. Now, the clumping, the agglutination has occurred. What this is going to lead to is a couple of things can happen here. One of the things that can happen is get this big, huge macrophage. That's going to do phagocytosis to get rid of that. Okay? That's one thing that's going to happen. But look what I have up there for 2. I have complement proteins as a mediator. So what do you think is another thing that can happen here?
Mac?
MAC is going to be formed. Exactly. So this can happen, but then the other thing that can happen is that you have this red blood cell, you form this antibody, antigen complex, and then you're going to have a complement protein come up here and acknowledge that on that cell, okay? That's going to lead to the complement protein cascade. So then we're going to eventually get C3, right? And then we're going to eventually get this complement protein that's going to start inserting MAC, right? Now what happens?
Sodium rushes in, H2O follows, and then lysis.
Sodium and water come in, and then eventually what this is going to lead to is this cell, this red blood cell, doing lysis, which then actually ultimately leads to a bunch of hemoglobin in the plasma. Okay?
That's the type B red blood cell lysing.
That's with any kind of wrong blood transfusion. I'm using as an example a person that is a type A recipient is given type B blood.
Right, but is an antibody antigen complex going to form on the type A red blood cells? Or
No.
Because there's no antibodies for it, it's only going to form on the type B blood cells?
Because that person with type A blood has those anti-B antibodies that's recognizing that B antigen is not self.
Okay.
Okay?
And with the agglutination and coagulation, there's no clots forming, pulmonary emboli.
That could also form as well, for sure. Definitely. You could have other complications, but the thing that actually ends up being the thing that leads to a person's death in this situation is shock, because you've ruined the kidneys, you've increased the pressure here, and those kidneys, those little capillaries in the kidneys start to just get destroyed, and you start to see bleeding out in those kidneys and blood in the urine.
What type of shock would it be?
I'd say it would be
Hypervolemic?
hypovolemic, I would say, because you're losing that blood in the kidneys from that circulatory system.
Okay.
Yeah.
So how would it choose which route to go?
What do you mean? Oh! Honestly, I would say that both of these are happening,
Okay.
actually. Mm-hmm.
So, because O is the universal donor, does O not have any specific antigens displayed on its outside?
It does have antigens displayed on its outside, but the antigens that O displays, our bodies, everybody doesn't recognize them as non-self, so they're not a problem. So when we're talking about blood typing, we really are only looking at the A and the B antigens, because those are pre-formed antibodies that, depending upon your blood type, you already have circulating in your blood. Okay, this is one example of type type 2 hypersensitivity. I'm going to give you another example that is very different, okay? But the key here is to acknowledge that type 2 is tissue specific, all right?
Can I just,
Yeah. Another example that I'm going to do is an autoimmune disease that's called Myasthenia gravis. Have you guys heard of that? Myasthenia gravis. So I'll write that down. Or myasthenia. Myasthenia. Now, a manifestation of this is muscle weakness, okay? So, manifestation is muscle weakness. And kind of think of it as the inability for, and I'm talking about skeletal muscle, right? The inability for skeletal muscles to contract. And what's really going on here in this particular one is you're going to have okay, here is my little picture. All right, and I have these striations going on. This is just the skeletal muscle, okay? Skeletal fiber. This here, this is what's called the motor end plate. You guys remember that? Okay, so this is the motor end plate right in here. And how does it normally function? What happens at this motor end plate? What goes on?
Acetylcholine is released.
So this motor neuron is going to have vesicles that contain that neurotransmitter acetylcholine. And as you have an action potential go down this motor neuron, that's going to, at this axon terminal, cause the influx of calcium. That influx of calcium causes these vesicles, ultimately, to fuse with the cell membrane and, yes, release acetylcholine. So acetylcholine is going to be released into this synaptic cleft here. And normally, what will happen is you're going to have all within this membrane here gated channels. Are those channels, what's making them gated? What has to happen for them to open?
Acetylcholine has to attach to a receptor.
Right, so these are,
Gated.
they're chemically gated because of that. They need a molecule to attach to them. So you're going to, for these receptors that are chemically gated, acetylcholine has to bind to it. And when acetylcholine binds to it, what happens?
It starts its releasing whatever.
It opens, right?
Mhm.
Let's say it opens, because it's gated, so let's just say it opens. Now, why did we do that? What is the purpose? Look, we have to think what's going on here. The inability for skeletal muscles to contract. I haven't yet said what's wrong yet. We're just looking at what happens normally, okay? But why are we doing this?
Depolarize.
To depolarize, and in simple terms, to excite this muscle fiber. So we can't have a contraction, because we need to depolarize this membrane, have an action potential along this muscle fiber that then goes down these tubes, right? And then that's going to have sarcoplasmic reticulum, SR, releasing calcium into the cytosol, right? And that's needed to start those chains of events for you to form a cross bridge, right? But the thing that's happening in this type 2 hypersensitivity, and why it's an autoimmune disease in a sense, is that this acetylcholine is blocked, and it's blocked by an immunoglobulin, an antibody. So you're going to have an antibody how about I do this? I'm going to write it as a different color and say, hey, here's our antibody. All right? This person that has this disease has already preformed antibodies that are essentially have enough of the molecular structure as acetylcholine to bind to these receptors. When that happens, you're not going to be able to have this muscle fiber get excited. Okay? So these antibodies essentially are blocking the ability for acetylcholine to bind to that chemically gated ion channel. And then this doesn't happen, and this doesn't happen, and that muscle will contract. Okay? So really get that one. It's a little bit different, okay? And in this particular one, it doesn't really have an effector cell here, right? There's no effector cell going on. Instead, you're seeing that the mechanism with this hypersensitivity is put into this group because it is the antibodies that are leading to this dysfunction of a person that has this, okay? So we're good with that. Now the next one that I'm going to talk about, that I don't have a picture for.
You said it's leading to infection?
No, this one's different. This one is a weird one.
Yeah. No, but your last word was, you said it was the antibodies that are leading to
Dysfunction.
dysfunction.
This is where this person, their skeletal muscles aren't going to be able to contract because they aren't able to get excited.
Right.
So because they're not being able to get excited, would that lead to atrophy?
Oh, for sure. Definitely. So, I mean, if you don't use something, it shrinks, right? When we're talking about stuff in the body, for the most part. I actually think hypersensitivity,
The book
too,
says the
has a
result.
lot of variation.
Sorry, the book says the result is self-regressed. I mean,
That's so you got to realize these people aren't going to die, you know? And it's not like every single one of these is going to get blocked. It's just that when somebody's exercising, you want them to be able to contract more, and they're going to get weaker faster. And so those individuals can come to normal functions for them after resting, after exercise. I personally would think that somebody that has this probably doesn't exercise that much anyways. And I'm not entirely sure of the whole pathogenesis, if it's something that ends up developing or if it's more of a congenital thing. I don't know all the details of it, to tell you the truth. It's just this one in particular, type 2, has a lot of different ways that it manifests. And the key to it is it's tissue specific, all right? Just another example, Graves. Are you guys familiar with Graves' disease?
I have it, whatever.
A similar
So-
thing is actually happening with that in the fact that you have an antibody that's gonna mimic thyroid stimulating hormone. And so this antibody mimics thyroid stimulating hormone and prods that thyroid to make thyroid hormone when it shouldn't be.
So they have to destroy my thyroid gland, I don't have one.
Yeah, and so then their thyroid will get goiter because that thyroid's making more thyroid hormone than it needs to, and it'll get bigger. And the thyroid hormone is about metabolism. So people with Graves' disease have an increased metabolism because of that, because they're secreting thyroid hormone when they really don't need to.
So Graves is hyperthyroidism?
Now hypothyroidism is.
Hyper.
Yes, hyperthyroid. But a type 2 tissue specific could be hypothyroidism, different mechanism. In this situation, you're going to see the body attack that tissue and start to destroy thyroid cells so they can't make thyroid hormone.
So in this one, where it actually would lock the channel instead of actually keeping it open, the opposite is true for like Graves, where it actually is stimulating,
Correct.
not blocking.
Correct. Yes, it's mimicking thyroid stimulating hormone and prodding that thyroid to make thyroid hormone when it shouldn't. Exactly.
So the opposite, if that happened in this, it would actually create spasms, et cetera, the opposite.
Yeah, if it did it that way, but it doesn't. Instead, it blocks you from
Blocks.
having this electrical event on this muscle. Other questions? I definitely, when you guys go to start studying this, because I think takes a little bit of time. And I think that getting out a blank piece of paper and seeing if you can outline it without looking at something, eventually, definitely not right away, will really help to get it down. And we're seeing a lot of stuff that we're familiar with. So if you come across something you're like, oh, I forgot that, go back to that Pearson book and look at how it normally functions to understand what's going on when it's not functioning normally. So I can erase this now. All right, so then the other one, the next one is called hypersensitivity 3. And now we're getting into hypersensitivities in general that take a little bit of time for them to develop. So now I'm going to put a 3 here. For hypersensitivity 3, talking about a persistent infection usually, but there is, are you guys familiar with lupus? That actually has a genetic component to it, just so you know. So it's a little bit different, but I'm going to focus on when there's a persistent infection, what's happening there. And this one is not tissue specific. And so what we're going to see in hypersensitivity 3 is okay, I'm going to write the infection now. Think of this as that non-self antigen, okay? So here's this non-self antigen. But a person can't get rid of it. It's lingering, all right? What we're going to see eventually with this non-self antigen, because the body is unable to get rid of it. So in regards to an infectious disease, what end part of that pathogenesis is never reached here? Resolution is never reached. This is a persistent infection where your body is unable to destroy it. It lingers around. Resolution is never achieved here. Body's unable to get rid of it. These things are lingering around. And think of them sort of as lingering around. I think the best way to think of it is they're lingering around in the plasma. Okay? And so, body unable to get rid of it, lingering in plasma. And what you're going to have in plasma is going to be antibodies, right? And the antibodies are going to start to latch onto it to try to get rid of it. So these are antibodies. All right? And as you start to form this antibody-antigen complex again, right? Form another antibody-antigen complex. But this particular one is going to precipitate out of solution. All right? So it was before dissolved in that water of your plasma. But now all these antibodies are tagging it, and it's going to go out of solution. All right? So you form this big complex here, and this whole thing here is going to precipitate out of solution. I'm trying to think of something, where you have this solution and then I'm not a big on chemistry, but you do something and then it starts to precipitate out.
So basically making like a solid?
Yes, that's what you're doing. This thing that was dissolved is coming out of that solution and is becoming a solid, if you will.
From an oversaturation, maybe?
It's doing that because you're getting all these
Binding.
antibodies attaching to it, and that particular thing actually makes it go out of solution. Now, as it goes out of solution, I spelled that wrong. Solution. Oh. As it goes out of solution, you got to realize this is in the blood. It's not tissue specific, and what you're going to start to see in various areas of the body build up of the antibody antigen precipitate. Okay, so let's look at it here. So let's say here's your tissue, here's this antibody-antigen complex, and it's going to precipitate out of solution. And what it'll end up doing is setting up house in these tissues. Starts to build up in these tissues. Okay? So it makes sense to everybody what I got down there. And it's not tissue specific, all right? So it starts to build up of antigen antibody precipitate in tissue, any tissue type that is precipitating out of solution, okay? So here's your tissue here. Now, what's going to happen next is you're going to see it in this type 3, because that's what we're looking at, right? The mediator here is complement proteins, right? The formation of this antibody-antigen complex and it going right here is going to trigger complement cascade. So this is going to, as it builds up in the tissue, you're going to see that complement proteins are activated. Does complement always lead to just lysing of a cell, or
No.
does it do other functions?
Could probably call in neutrophils.
Right. Complement promotes inflammation, and it's going to, when we think of inflammation, not only are we having vasodilation occur, vascular permeability, but yes, we're saying, come on over, neutrophils. Because C3 here, our effector is neutrophils. They're going to get called to the scene here. So complement proteins are activated, and inflammation occurs, and neutrophils migrate to the site where we have this buildup occurring with this persistent infection. So we also have to keep it in mind. So now a neutrophil is going to come along, right? And that neutrophil, what it's going to do, but a neutrophil, here comes a neutrophil. Neutrophil. And that neutrophil is going to, is a neutrophil a granulocyte or an agranulocyte?
Granulocyte.
Granulocyte. So it has a lot of these granules as well. And it's actually going to start to release enzymes from those granules, okay? So this neutrophil is going to start to release enzymes from its cell through, exocytosis. And these enzymes are going to start to break down our cell's tissues. Enzymes break down tissues. And I mean, you could have anywhere in the body that this could occur when you have a persistent infection. But the most common place is going to be the kidneys. Because it's the kidneys where we're trying to get rid of this, okay? Trying to get rid of the toxins and stuff in our body. And so when this happens, you start to see inflammation of those capillary beds in the kidneys.
How do free radicals play a part in this?
Oh, okay. I'm imagining that the neutrophil, anytime you do metabolism in the body, you form free radicals. And so I'm imagining just the normal metabolic process of that neutrophil making these enzymes, it's made free radicals that also get released. And free radicals are going to want to make a bond with anything. And it ends up being an abnormal bond that then leads to something not functioning properly. And that's just a general
Okay.
view of it. I don't know any specific details of how one particular, except for the one that we talked about with that detergent agent that we did way back when, where they used to have that one molecule that was a detergent agent, and not detergent agents, dry cleaning.
Oh, yeah.
Yeah, the dry cleaning product, which I guess they don't use anymore. I have no idea.
Hopefully.
Hopefully not, right? But that was an indirect sort of free radical response that ended up leading to, I believe it was damaging liver cells, if I'm thinking correctly. We're going to stop here, and then we'll start on hypersensitivity 4. On Thursday, I do want to remind you that the second exam for this class is going to be now March 16th. That's a Thursday. It's not going to be next Tuesday. All right? We're going to start in on white blood cell malignancies as well, and hopefully we have at least 45 minutes on Tuesday to have a review session for exam 2.